everyone, my name is Michelle Moyer. I'm a PhD student at the University of Maryland, Baltimore County, and today I'll be telling you about female and male song in Orchard Orioles. So historically, many elaborate advertisement traits in animals, such as vocalizations or plumage, have often been assumed to have evolved as a result of sexual selection acting primarily on males. And the study of birdsong as a whole has been dramatically impacted by this long-standing bias towards male traits. This can be seen just in who has been studying song in the past. It's been dominated mostly by men in North America and Europe, and more diverse researchers, especially those in the tropics, have really grown the field and demonstrated that female song in particular is more widespread than initially assumed. Karen Odom, a former lab mate, and her collaborators released a groundbreaking study in 2014 demonstrating that female song is most likely the ancestral trait of songbirds. 71% of the species that they investigated had female song. And so as the field of female song is growing and as our knowledge base is expanding, there's a call to investigate female song in temperate and tropical species, particularly in species that have been well studied in the past where female song may have been overlooked. One such species is my focal species, the Orchard Oriole. They're a migratory temperate breeding blackbird, and they're found across the eastern US and into Canada during their breeding season. Males and females are dimorphic, so they look different from one another. There's a male there on the left and a female on the right. And there have actually been several mentions of female song in the literature on this species, but these mentions are mostly brief and anecdotal, and there has never been a detailed analysis of female vocalizations in Orchard Orioles. In fact, our lab has been studying this species for over 5 years, but it wasn't until our research on female song in tropical species prompted us to look for female song in temperate species that we first noticed it in the Orchard Oriole, which really highlights the need to re-explore previously well-studied systems, as I mentioned earlier. So this was the first intentional detailed analysis of female song in this species. I had two main research questions for this project. The first is just how often is each sex singing? I recorded individual male and female Orchard Orioles during their breeding season and calculated their song rate as how many songs they produced per minute. I then took these recordings and used Raven Pro, which is an acoustic analysis software, to perform detailed analysis of 9 different song characteristics on 3 songs from each bird. The goal for that analysis is to determine if males and females are singing similar songs or if females sing something completely different from males. Here are the results of that first element, the song rate analysis. We have time in the x-axis and song rate on the y-axis. The reason that we're plotting this against time is because we wanted to see if there would be an effect of time on the song rate of either sex, or if their song rate changed throughout the breeding season, such as when incubation began or after their chicks had hatched, etc. We have female song rate on the left, male song rate on the right, and there's obviously a pretty clear difference here. Males sang a lot more frequently than females did. On average, males sang about 2.35 songs per minute, where females sing only 0.13 songs per minute. However, females do sing quite consistently as a whole. Of all of the females that we recorded, 59% of them sang at least one time, so this is not just an isolated, rare behavior, and they also sang well into June. So now I'll walk you through the acoustic variables that we investigated. For those of you who aren't familiar, this is a spectrogram, which is essentially just a visual representation of a sound. It's like reading sheet music from left to right. We have time on the x-axis and frequency on the y-axis. And using these spectrograms in Raven Pro, I investigated the duration of the full song. And songs are made up of song elements that we classified as syllables, which were just elements at least 0.01 seconds apart. So I also looked at the duration of syllables and the syllable rate. I also looked at the proportion of pause, which is essentially a metric of how much silence is in a song or how much silence is between the syllables. I counted the number of syllables. I looked at minimum and maximum frequency and the difference between those, which is the bandwidth. And also, Raven Pro produces these traces of the spectrogram, which you can see there in that neon green. And an inflection point is when the frequency changes from either increasing to decreasing or vice versa. So it's essentially just another metric of the variation in a song. Before beginning our full analysis, we ran a correlation matrix with all of our variables, and we found that full song duration and number of syllables were highly correlated. They had a correlation value above 0.8. And so we actually decided to remove the number of syllables from the final analysis to avoid measuring any redundant elements. Furthermore, 4 of our variables that you see here underlined did not meet the ANOVA assumptions of equal variance, and so they were transformed to meet those assumptions. We also removed several outliers from minimum frequency for it to match in normal distribution. We did run a non-parametric test on these 4 variables to make sure that the results of the parametric test were consistent and were not affected by our transformations, and we did find that they showed similar results, as I'll show you later. We believe that this was the best way to handle our data, but if you have any other suggestions, I'd love to talk with you more. So before I jump into all of our results, I'd like to tell you what they actually sound like. So here's what a female Orchard Oriole sounds like. And here's what a male Orchard Oriole sounds like. So these spectrograms are showing female songs on the right and male songs on the left, and there's already one clear difference here, which is the duration. So females on average sing songs that are 1.26 seconds in length, while males sing songs that were 2.84 seconds in length. And this difference was significant. So as I mentioned before, we ran an ANOVA. We actually ran a nested ANOVA with individual nested within sex to account for within sex variation, as I'll show you here. So full song duration, as I mentioned, was significantly different between males and females, with males having longer songs. Females, interestingly, had longer syllable durations. Males had a higher maximum frequency and a greater bandwidth. And males also had more silence in their song, or a greater proportion of pause. However, I'd like to highlight here that the within sex variation column, which is that column all the way there on the right, was significant for all 8 variables, which suggests that there's a lot of within sex variation. Females differ a lot from other females, males differ from other males, etc. So one of the next steps for this analysis is I'm planning to calculate the coefficient of variance for each individual to see if there's a difference in how the sexes vary. As I mentioned, all of those variables that we transformed were tested using non-parametric tests as well as the ANOVA, and all of our tests showed similar results. So the big takeaway that I'd like you to have from these results is that males and females were significantly different for 5 of the 8 variables that we studied. The majority of the variables that we investigated, they showed statistical differences in their songs. And so the obvious next question is, does this difference matter? Can males and females tell each other apart based on song? Do they care if they can? So this past summer, we actually completed live playback experiments where we played male and female song to Orchard Oriole pairs and observed their responses. We hope that analyzing these playback experiments will help us to develop potential hypotheses for how or if female song functions in this species. I also plan to study a closely related tropical species, the Bahama Oriole, to develop hypotheses about how female song may function in a tropical species. In general, this study really highlights the need to re-explore well-studied species where elaborate traits such as female song may have been previously overlooked. Thank you very much for your time, and I look forward to taking any of your questions.